ABI Tuesday seminar. Today we have Dustin Williams. He is an associate professor in the Department of Orthopedics at the University of Utah and a research scientist with the Department of Veterans Affairs. He has BS degree in microbiology and PhD in bioengineering, and he fuses these two to develop antimicrobial technology that targets biofilm phenotype. He has launched 2 companies already, and so he combines this translational science with commercialization to improve patient care, reducing rates of biofilm-related infections. So today we will be hearing a lot about this biofilm, so we look forward to your talk today.
Oh, thank you very much. Great, well thank you, and this is just an honor. This has been such a fun trip. And I should really quick here give a wave hello from my kids. They said to tell everybody hi from Utah. So we got 5 kids waiting for us, and we're excited to get back to them, but this has just been so great to be here. And all the way from 6,000 miles away, they said to say hello. So I want to talk a little bit about biofilms. First of all, I'm guessing this group knows quite a bit about biofilms. Raise your hand, please, if you would. You've heard about biofilms. You know what they are? Okay. So we won't get bogged down in all of the background behind them. So I am going to disclose that I have 2 conflicts of interest, Curza and Pergo Scientific. I'll talk a little bit about both of these companies and the technologies that we've developed that go along with them. But let me kind of begin by talking a little bit of just a couple of problems that we're interested in fighting. And sorry, I forgot to give warning of gross pictures. I get in trouble every now and again. I have to give warning. I'm about to show gross pictures. And so, I mean, this one's just insane. The amount of pus and whatnot coming out of a periprosthetic joint space. These actually are pictures you can find online. This is, antibiotic-loaded bone cement beads that are packed around a total knee implant. And this problem of open fracture and periprosthetic joint infection has gotten so bad that we're essentially now taking Parmesan cheese and pouring it into a joint space. Hey, what else do you got? Straight antibiotic powder. Just dump it straight into the site and hope it works, right? That's the state of affairs that we're at. Orthopedic surgeons really don't have any other answer at this point. And so we're hoping to help in this fight. So if you look back at the history of open fractures and prosthetic joint infections, for about 40 years now, we've had no change in rates of infection. They're still the same as they were. When Gustillo defined open fractures, wounds, about 35 to 50% for type 3B open fractures, still the same today, what we're looking at. Now, you'll oftentimes hear people say 15%, 25%. What's reported and what's actual are very different. Jay Parvizi talks about this a lot in his talks. Another area of interest for us is chronic wounds. So we work at the Department of Veterans Affairs. A lot of veterans, a lot of diabetics who come into the clinic. And about 80% of the foot clinic in our VA at Salt Lake City, Utah, is dealing with these diabetic foot ulcers. And very problematic. They recur. The amount of recurrence is very high. And getting healing and curing, very low. So amputation is a very real thing that has to be faced. And I think we're all familiar with that. Okay, so kind of talking about biofilm materials-related infection, biofilm implant-related infection. We have a couple of seminal papers that were published. You may be familiar with these. 1982. We have a 56-year-old male had an elbow abrasion while working in the garden, not too far distant from the face scratch that a police officer got in New York City. First patient who received penicillin, right, had an infection from a cut in the garden. Elbow abrasion, he had had previously a pacemaker implanted with a lead, and after the elbow abrasion, he began to have pain in his chest. And so we went on these multiple courses of antibiotics. He would get better. The antibiotics would be taken away, get worse. Get antibiotics once again, get better. Antibiotics taken away, get worse. So you begin to see this recurring cycle of infection. So this is one of Bill Costerton's early papers published on a biofilm material-related infection. 1984, same type of scenario with a long bone intramedullary nail in the femur. So on this one, this patient, I'll have to look at the paper to remember the specifics, but was on antibiotics on and off for about 7 years. That's quite a long time until they went in, take the implant out. Bill Costerton and his group imaged the biofilms on there. Once the implant's gone, a final course of antibiotics, patient's healed up. Same thing happened with this previous case. Once they took the pacemaker lead out, identified biofilms on it, and then a final course of antibiotics, healed up. So once the foreign body was gone, healed up after that. So this is a very common, kind of the hallmark cyclic nature of biofilm implant-related infections. Getting better, getting worse. My father recently retired from the Air Force in the United States, and one of the last meetings he went to was actually talking about biofilm-related infections. And they asked, well, what do we do? And he said, all they told us was hit it fast, hit it hard. Nobody else has any answer, right? We're still in this room today because we don't have the answers. We're trying to look for them. Got a lot of questions. So the key player behind all of this is biofilms, as you all well know. These resistant, recalcitrant, core persister cells that cause these problems. I like the work of Claude Zobel. Oftentimes we recognize Bill Costerton as the father of biofilm discovery and research, but he kind of built upon many observations that were done before him. And Claude Zobel was one of those. So he was an Idaho boy. So another famous Idaho boy, right? So Idaho's right above us from Utah in the United States. Philo T. Farnsworth invented the television. He was from Idaho as well, and he saw these lines from the plow, and he saw this cross sections and how he could make a picture. Quite fascinating. Claude Zobel, he wanted to discover things, so he went to the Scripps Institute of Oceanography. And he began to observe that bacteria collected from seawater would preferentially adhere to the surface to the hard surface rather than stay floating in the liquid, the seawater. And so that was his kind of premise, and his teaching was bacteria prefer to stick to hard surfaces, and they coalesce and congregate together. And now it's understood that about 99%, or estimated that 99% of bacteria in natural ecosystems dwell in biofilms, not in the planktonic or free-floating state. Now that's also including human skin, right? So I apply this to think a little bit outside the box when we consider a surgical approach to implanting devices. Oftentimes we take incredible care to filter the air, right? We put mupirocin in our nares to get our MRSA bioburden down. We scrub our entire bodies. We use laminar flow systems. But what we often don't account for is the very bacteria that's dwelling deep down in our skin. So Nick and I actually have recently published a book, and a couple of our chapters touch on this concept. And so you think about in a surgical procedure, I go in and I prep the top 2 to 3 layers or so of the stratum corneum, but the bacterial biofilms, for example, C. acnes, Propionibacterium acnes, now Cutibacterium acnes, they renamed it. Microbiologists are just annoying that way. They're just renaming stuff all the time. You got to keep on top of it. C. acnes can dwell deep down multiple layers in the sebaceous and hair follicles, sebaceous glands and hair follicles. So when I take a scalpel, for example, and then I cut the skin, I can actually push my own flora microorganisms that live deep down into a surgical site to colonize an implant. And this work's been done, I believe, a study out of Japan where they looked at pacemaker implantation procedures. They cultured the site every 15 minutes and watched C. acnes numbers climb throughout the procedure. Within about 30 minutes, the C. acnes bacteria will already begin to recolonize a cut in the skin. So our own source of biofilms could be contaminating implants. So I think I share this in a bioengineering context to think about when we're developing medical devices, think of the entire paradigm of the environment that it's exposed to. Oh, okay, so we have to remember, right, if you take nothing else away from my talk and my visit to New Zealand, I've heard you have penguins here. So we have to share the whole gist of all of this is ultimately, if this is biofilm and this is us, to just kind of take down the biofilm, right? Very simple message that we have. So that's the goal that we have. So biofilms, and this is thanks to Nick for this slide here. biofilms are really kind of fighting against us with two major components, and that is they frustrate phagocytic engulfment and they resist clinical intervention, right? So I'm going to show a little bit in just a bit about our models and how if we put a biofilm into an in vivo space at the time of surgery or trauma, we are instantly recalcitrant to antibiotic therapies. So kind of to dwell upon this a little bit more, again, thanks to Nick for this, some of the early experiments we actually heard on Friday about this, the ELEC experiments, where they took and they could see that if you have a foreign body in the skin, your infectious dose can go down by 10,000-fold, which means the bacteria can have something to find and stick onto and cause infection at a much higher rate than if there's no foreign body present. The ELEC experiments were a little controversial, and that indicates just how much faith the world had in penicillin. They actually did these experiments in medical students, right? So to be able to graduate, you had to be willing to have Staph aureus stuck into your forearm. They don't do that anymore. I don't know if you've heard. So, but if a biofilm is essentially larger than a neutrophil, the cluster of bacteria becomes about 10 microns, it becomes very difficult for neutrophils to attack or swallow them. Phil Stewart, for whoever was there on Friday, he actually showed videos of this, the difficulty of neutrophils engulfing bacteria in these even microbiofilms or microaggregates. Okay, so as early as Alexander Fleming's discovery of penicillin, he already began to develop this MIC, or minimum inhibitory concentration panel. This ability to use dilutions of antibiotics to see just how sensitive bacteria were to antibiotics. But I'm going to touch more on this in just a moment. In the biofilm phenotype, bacteria do not behave the same way they behave when they're in the planktonic state, when they're single-celled organisms. And just to kind of highlight this, you've probably all recognized this, but to highlight it even more, this is all, again, based on Nick's work here. So Nick has been a postdoc. He's actually going to be faculty in like 4 days. He's promoted to faculty. So this is his work from our lab. You know, we have this MIC panel, so the amount of antibiotic that can inhibit bacterial growth in a test tube does not necessarily correlate with how well it's going to attack these biofilms. So Nick has taken even vancomycin up to its solubility limit of 64,000 micrograms per mil, and the number of bacteria in these biofilms still hovers as though they were untouched. We can essentially throw an atomic bomb of antibiotic against biofilms, and they're happy to just live in it. Now, there was a paper just published on mold growing in Chernobyl, so it can actually withstand amazing things. Biofilms are amazing. So we recognize these two different phenotypes, biofilm, planktonic, and they behave very differently. Yet everything right now in a clinical reference laboratory, everything that's regulated by the FDA, 100% based on outcomes related to planktonic bacteria, right? And I think that's why we're seeing some disparity in clinical products. So we still, to this day, are using traditional antibiotics the way they've kind of always been used. We are applying them to coatings, right? My kids used to suck on the grocery cart handles. Triclosan was put into plastics a lot, right? It's no longer allowed in most plastics, in hopes that we could get a little bit local high-dose concentration of antimicrobials to resist this biofilm growth and formation. But oftentimes what we don't realize is all of our finger oils get onto these bars here, and they'll grow just fine, right? Because you give a little coating onto it of your oil, and then the antibiotic coating doesn't do anything. So we see a lot of these common agents being used: silver, gentamicin, triclosan, active release, passive release coatings. So essentially, everything works in vitro, some stuff works in vivo, and hardly anything works in clinic. That's what we're finding. In fact, when Nick and I were sitting in Washington, D.C. about a year ago, in November, and the exact same thing was found in wound healing studies, implant-related infections, people doing clinical trials. A lot of the urinary catheters that are coated with chlorhexidine, with silver, they really aren't performing as well as anybody wants them to. And so still, we have companies constantly calling us, asking us, how do we solve this problem? And I ask them, well, don't you have silver on the market? Well, we want something that works, right? There's still a problem. It's not doing the job clinically. So we control wonderfully well on bench tops, all of our experiments, right? But once we get to the clinic, it's a whole new ballgame. In fact, we sat down with surgeons a few months ago, developing a medical device that I'll talk about here in just a bit. And he said, there's a lot of things you don't take into account when you're developing something on the benchtop. For example, a lady comes into my office the other day and says, my cat won't stop licking my catheter. So, you think about, what are they being exposed to? So a catheter wound site, right? A catheter site could be licked by a cat. So you got to take these things into consideration. There's a few things, even in the lab, that I think we can take into consideration that might help us improve our outcomes on the back end. So as we think about clinically what's going on. One of the things I've been kind of preaching for years, if you will, you'll even see in my early publications, is if, like for example, if we're going to develop an active release coating, we have to take into account dynamic flow of fluids. So most of the research you find on an active release coating, people will take a product and they'll put it into a stagnant broth solution and they'll look at the elution profile of an antibiotic over time. Maybe they'll take a sample once a day for 7 days or something, right? But this is sitting in a stagnant broth solution. The amount of antibiotic is continuously increasing above physiologically relevant levels. But if I put a flow into that system, then I take into account this dynamic environment that the body is continuously exposing our implants to. I have consulted with company after company that makes billions of dollars, and I've tried to drive this home. You can't do all of your tests for a coating in stagnant broth. It has to be in a flow system. Because what's going to happen is you're going to have all of these false positive results. You're going to get wonderful antimicrobial activity in a stagnant broth that does not translate to the human condition. So a lot of my early work was based on that. Also, MIC values do not correlate to antibiofilm activity, right? We've been driving this home. Nick's talks, his message, our chapters, we're driving this home. Everything is based on a clinical reference laboratory, a MIC susceptibility panel. It does not correspond to how a biofilm. Oh, that's the minimum inhibitory concentration. So that the Alexander Fleming picture I showed where you all of a sudden have this amount of antibiotic that can inhibit bacterial growth. But if a biofilm is there, they have a different characteristic or phenotype that resists the antibiotics.
And it's important that that's used clinically to prescribe antibiotics.
Exactly.
And it's also required by the FDA for med device testing.
Exactly. So again, it can produce, I think, many false positives. Okay, so the other problem we run into is lack of sustainable release and using biofilms as initial inocula in our models. I'm going to get to that in just a bit here. Okay, well, right here. So one of the approaches, the unique approaches that my lab takes, and I began to propose this as a graduate student with the help of clinicians who were in a lot of discussions with us. How do we model things more accurately? So one of the areas I was interested in was this area of open fractures. So we have a lot of soldiers who have this part of the tibia, the proximal medial aspect of the tibia, is one of the most susceptible areas for breaking an open fracture. And so a type 3B open fracture is one that has a very large wound, periosteal stripping, and high doses or amounts of contamination. Now, if I consider this open wound occurs, let's say I have a soldier running in a mud river or out in the dirt, they could break open their leg and immediately scrape it into the dirt. Now remember that 99% of bacteria already dwell as a biofilm in natural ecosystems, including dirt, mud, or soil. So I take this, traditionally we look at planktonic bacteria as adhering to a surface and growing into a biofilm. I take that and I flip it and say if I have an open fracture, it already is contaminated at the time of injury with a well-established biofilm. So if I give IV antibiotics to a patient that already has a biofilm there, I might be lucky to get 50 micrograms per milliliter of antibiotic in my blood levels, but that's not enough to kill the biofilm. So it could persist. Once the prophylactic antibiotics are taken away, then that biofilm could flourish and grow, right? So some of the ways that approaches that we take to target biofilm implant-related infection are using biofilms as initial inocula. We take this dilution into account in the body. We're developing unique antibiofilm compounds that specifically eradicate biofilms, agents that attack core persisters, and sustainable release technologies. So I'll show just a few of these, and this is just an example of one of the things early on that I was working on. And that is, let's say I have these biofilms that contaminate a bone surface at the time of injury, then I could develop a coating for a fracture fixation plate that could release this antibiofilm compound into the space, interact with the biofilms that are there at the time of injury, and then I could effectively create what I call Staph aureus potato chips, right? Because the compound that we use deflates the cell by puncturing the cell membrane and disrupting the biofilm. So some of the compounds we're developing have this ability, this dual ability to both disrupt and kill bacterial biofilms. In this particular model of developing an active release coating, this was actually all of my PhD work was based on exposing this coating to dilution or to dynamic flow. So I would take and I would grow well-established biofilms, put them next to a simulated fracture fixation plate, connect that to a piece of bone, and put it into a flow cell system where flow would be introduced to the system at the rate of interstitial fluid flow in the body. And then I would optimize an active release coating that way so I could really, really target how much antimicrobial do I need to load into a coating to eradicate biofilm, not just planktonic bacteria or not just inhibit growth of planktonic bacteria. So in this particular model, I used two simulated fracture fixation plates in the proximal medial aspect of a sheep tibia. And I used two plates so that one can be used for microbiology and one for histology. So I can leave one in place to look at what's happening with the immune cells and the biofilm in that space and how is the active release coating affecting the tissue. The other one for microbiology. In this particular model, we got 100% of our animals that did not have an active agent to kill biofilm, 100% were infected. That's actually important because a lot of animal models are challenged, right? They don't always produce 100% infection 100% of the time. And so this was actually a pretty big advance, using biofilms as an initial inoculator to get this very strong osteomyelitis signal. Whereas those that we treated with our active release coating, 100% were infection-free, which was a big success and indicating this ability to translate through and target the biofilm phenotype. So this is one of the models we've developed along these targeting lines. Another model I wanted to kind of just share briefly is a model that we've just recently developed and which was a significant challenge to develop, and that is a large animal model of heterotopic ossification. We wanted to develop this model because 62% of soldiers coming back from Iraq and Afghanistan who are hit by an IED blast and have an amputation, 62% develop heterotopic ossification after this blast-related trauma. So that's bone that's sprouting off the normal regions called ectopic bone, growing into those regions where it shouldn't be growing. So we believe that the osteoblasts are kind of confused. What do I do? Growing into spaces where they shouldn't be growing. So just briefly, this is actually a sample of heterotopic ossification from a patient. We actually took 40 of these samples from soldiers who had heterotopic ossification at Walter Reed, and we wanted to first identify and understand what does this bone look like. Long story short, it's a mixture of cortical and cancellous bone. So it's beginning to have this kind of confused, what am I doing, where do I go? It's a confused bone. So we wanted to develop a model that could mimic this or that could mimic that same outcome. And so what we did was develop a sheep model, but recognizing if I have a blast-related injury, right, I'm going to have a variety of factors that hit me and affect me. So if I'm hit by an IED blast, I'm not just hit by a blast. I have bone chips that form. I have periosteum that's disrupted. I have sand that enters the site as well as biofilms that enter that site. So with this model, we took all of these different factors into account exposed sheep to these factors, including, I want to kind of show you, this actually took, to get approval to use this air cannon device on these sheep legs, took us about 16 months to get through the IACUC process. You call it the animal ethics process, right? Took a very long time. Very challenging model, but we were very excited that it was successful. So this is showing a silica bead to replicate sand in that environment. So we would take these silica beads, grow biofilms on them, and put those into that blast site so that we could mimic the natural state of bacteria, right, going into the body. This is some of the histology. I simply put this slide up to brag about our histology abilities, right? So Tyler Epperson in my lab would be so happy and proud to stand here and tell you how much he loves doing this histology. But we get, next to these biofilms on those beads that I just showed you, we get this sequester formation. We get this osteoclast activity, osteoblast forming the new bone, but really showing this combination of cortical and cancellous bones. So this model, using these biofilms as initial inocula, all of these multitude of factors to mimic a blast-related traumatic scenario, we were able to successfully achieve heterotopic ossification in 62% of animals, which corresponds directly with what we're seeing in the soldiers, 62% of them getting HO. So again, indicating here this growth of bone in the ectopic regions and the bone healing up where we drilled holes into the bone to release medullary canal components. So that's, I just kind of wanted to show some of the models that we're working with to develop and using biofilms as initial inocula. One of the really important factors of this model of HO was that even though we put biofilms into that site of the midshaft of the femur, these sheep did not require antibiotics. So that's kind of another phenomenon we're finding in our models is when you use established biofilms, put them into a wound site, you don't need antibiotics to help treat the infection because it's so low-lying and dormant and quiescent, which is in stark contrast to models when we use planktonic bacteria as initial inocula. You'll get raging and acute infections that develop almost immediately and require antibiotic intervention. In one of our models where we used planktonic bacteria, all of the animals had to be euthanized within 10 days because they get too sick so quickly. But the biofilms don't cause that problem. They can last out to 12 weeks without any problems. Okay, so one of the technologies we developed is with Kirza. And so these are great collaborators and friends at the University of Utah who've helped develop a new class of compounds that we use to specifically target the biofilm phenotype. And this gives you a flavor of one of those compounds. And this is actually one of the ones that Nick and us have published on and tested its efficacy in an active release coating against these bacterial infections. In this particular case, prosthetic joint infection. So this particular compound type is of that class that disrupts cell membranes. So we can see we got healthy kind of cells here. This is atomic force microscopy that we've done with these CZ compounds, we call them, with Kirza. And we can essentially pop the cells with these compounds, disrupting that cell membrane. And I suggest that if we want to think about eradicating biofilms, we need compounds that are nonspecific and globally interacting with the cell membrane. That's tough because our cells also can be affected by them, right? So finding a balance is important. But they do have reduced risk of resistance development. So one of the benefits of these Kirza or CZ compounds that we've developed is their dual ability to both disperse and kill biofilms. So on here we've grown biofilms of MRSA on the surface of titanium discs. This disc was exposed to water. This disc was exposed to our CZ compounds. And you can see how this is over an hour period we're able to effectively disperse those biofilms away from the surface. We've had independent analyses done on this as well with very promising outcomes. And for example, I mean, we could envision applying these to a lot of industrial sectors as well, not just medical applications. So depending on toxicity, balancing that out with the ability to eradicate biofilms on pipes, for example, could be very beneficial and promising. So this is one particular model in which we've used the CZ compounds in an active release coating. And this model was an extremely challenging model. We've just recently published on it, but you can see how well we can produce this osteomyelitis and prosthetic joint infection in that sheep. This is a sheep condyle in that space. I'm going to jump over this because we're getting short on time. But again, I wanted to just kind of show some of the histological approach that we take to analyze the effect that either a coating can have or biofilms can have on this bone in these regions. So this sequester formation is kind of that difficulty, right? Thinking about biofilm-related infections, this is the challenge too. An avascular bone chip where bacteria are using it as a nutrient source, but you can't get antibiotics to it. So that's where an active release coating is promising. It can elute right into that local space where vasculature might not be able to deliver antibiotics. So we envision being able to apply these CZ compounds into a wide variety of spaces and applications, which if you're developing antibiofilm compounds is always a problem. There's so many places you can put it into, but you have to kind of focus down and narrow it. So Kirza, these CZ compounds can be applied into, like I just mentioned, a lot of spaces. One of those spaces too is in biofilm-related wound infections. So I talked a little bit about diabetic foot ulcers. So we're working on developing a pig model where we utilize these biofilms as initial inoculant. And what I want to draw your attention to, so in one set of wounds, we'll inoculate these wounds with planktonic bacteria. In another set, we will inoculate with biofilms. We grow our biofilms on the surface of collagen, so bioresorbable collagen. And the reason we do that is because I want to be able to have the ability to inoculate, but I want the biofilms to stay behind and not the foreign body. So we grow on collagen and put that into the wounds. And in one of our models, in particular with Acinetobacter baumannii, we found in those wounds that were inoculated with planktonic bacteria, the body could essentially wipe out the cells, no problem. But in those wounds that were inoculated with biofilms, there were still about 5 million cells left in each of the wounds. So this ability of biofilms to hang out behind in the wounds is interesting and could help us identify, right, antibiofilm agents, not just antiplanktonic agents. Now, another interesting thing we found is even giving a 2-week course of colistin and imipenem in these pigs, there were still residual bacteria in the wounds. But they were down at a level of about 100 cells, or 300, 200, 300 cells, right? So even giving IV antibiotics, although the infection had gone away, there was still residual bacteria in those wounds. I talked with Don Granger, right? He works down the hall from Nick and I. He's an ID infectious disease doctor. He's been working on these diabetic foot ulcers in patients for the better part of 30 years. And when we talked with this, we talked with him about this, and he said, that just makes total sense to me. I see patient after patient who I give multiple courses of antibiotics to, and it just keeps on coming back, right? And so he thought, you know, the combination of IV with topical products could be a much more promising approach, right? A lot of podiatrists and foot doctors, they do that. Okay. So another very interesting finding that actually came out of Nick's work as well. So a combination of us developing this medical device I'm going to talk about in just a minute. He was trying to identify a compound that would work more effectively against these biofilms in our 2-plate model. And so he was working through a bunch of different compounds thinking, okay, how can I target these biofilm phenotypes? He identified a compound that has a miserably poor MIC value, minimum inhibitory concentration value. A doctor would look at this and say, oh, gentamicin, phenomenal MIC value. Gentamicin, phenomenal MIC value. And say, yeah, I'll choose that one to apply to my patient. A doctor wouldn't look at an MIC value of 2,500 micrograms per mil and say, oh yeah, I want that one for my patient. That's going to do the job. Never going to happen. But what we found is this particular compound, it did better against the biofilm than it did against the planktonic bacteria. So this particular compound effectively can kill the persister cells in the core of a biofilm more effectively than it can kill these rapidly dividing metabolically active cells on the exterior portion. And when you combine it with traditional antibiotics, you can kill both of the phenotypes in a biofilm. To the best of our knowledge, this is the first time that this has ever been seen, and we're very excited about it. And Nick will be publishing it in like 2 weeks. Maybe not. It's mildly toxic. Yeah, so this is a very exciting one.
It can be systemically dosed at about 50 micrograms per mil.
Yeah.
So these concentrations are still considerably higher than what you could safely dose, at least in this test. It's 24 hours. We're currently running time kill curves with this. The other thing is, I think Destin's getting towards this, it is below, we're seeing efficacy if you look at that cyan blue trace, which is the combination of gentamicin and that compound. It means that you could come up with local therapies, ways of release. And right now, we use stimulant. The doctors use off-label used, like antibiotic-loaded bone cements, things like that. So it could be a prime candidate for those kinds of therapies. It's not going to once it's released from area, it's not going to be systemically toxic because it would be concentrations that are tolerable. But it could be used for local therapy. Yeah.
Yeah. Yeah, so we're very excited about that one and the ability to use it, for example, in many of these spaces where we get these very dense biofilms. We've just published work. Surprisingly, in the literature, there's not a whole lot of data about the actual height of plumes that biofilms make. We just published a paper on it, and now we've been applying this to those biofilms, making sure they're nice and dense, and then testing its ability to eradicate those persister cells. But yeah, this particular compound, especially in combination with other antibiotics, performs very well compared to antibiotic products that are on the market in use today, right? So always the goal, how do we improve, how do we target this biofilm phenotype? So one other technology I want to talk about is an idea. So I worked for a long time in this space of active release coatings, and I still think these active release coatings have their application. They have their space to be in, but they're not going to solve all of the problems. We absolutely have to take a combination approach to the biofilm problem. There's no silver bullet. So I had worked in this active release space for a long time, and I recognized, first of all, to get an active release coating through the FDA takes a tremendous amount of time, a tremendous amount of money. And also, even if you got an active release coating approved, can't use the word approved, cleared, right, through the FDA process, you would only have it cleared for that one product. And then you would have to develop it for every other product you wanted to use it for, right? So that was a limitation that I said, hey, I would like to not have to do that. In addition, if I take and put vancomycin into a coating or gentamicin or whatever it might be, I'm locked into that antibiotic with that coating, right? I can't switch out my antibiotic. This was actually a question that came up in an AO meeting recently about the implant-related infection problem, saying we've put our beads in there with gentamicin. The problem is we cultured an organism that wasn't susceptible to it, and I can't switch out my antibiotic. That's a problem, right? It's a clinical challenge. So I wanted to develop a technology, a device that could get around these issues, make it through the FDA process, but also be applied to a variety of applications. I also wanted to develop something that could sustain indefinitely antibiotic release in a local environment, right? So a lot of the technologies that are used in orthopedics right now, for example, vancomycin powder, within about 12 hours, vancomycin powder that's dumped into a wound site is almost diminished. Within 24 hours, it's essentially cleared. If I put bone cement into that space, PMMA beads, stimulant beads, within about 3 days, 99% of the payload of antibiotic is gone, and I can't reload it. So we've developed a device that can release antibiotics essentially indefinitely into a local space. Right? Let me see if I have, oh dear, I did not undo the picture of the device. I can actually, probably just a minute. So with this device, we took our sheep model with the 2 plates. We put biofilms underneath those plates, which I've talked about. And we tested this in 5 sheep with the implant and the therapeutic into the device. We also tested against 2 sheep. Now these are very low numbers to this point, but we've just received a DoD grant to expand these numbers drastically, which we're very excited about. But we took 2 additional sheep with these implants and no therapy, so just the biofilm only, and also 2 sheep with the implants and biofilm, and gave them IV prophylactic antibiotics. So the thought is, right, if I give prophylactic antibiotics, I'm going to eradicate the bacteria. But I hypothesized that if I inoculate with biofilms at the time of injury or surgery, that they will not be affected by IV levels of antibiotic. Okay? So we gave gentamicin and cefazolin over a 48-hour period in 2 of the sheep. So the device was in that place for 10 to 14 days with the therapeutic, and then we took it out and then let them run a little bit longer to see if the infection got worse or progressed. Okay, and now in each of these sheep, biofilms were inoculated in those little membranes I showed earlier underneath those plates. So there's about 3 billion bacteria underneath each of those plates. And the reason I highlight that is because this is an extremely challenging model. I mean, you're asking a lot of your device or your antimicrobial to be able to eradicate the biofilms in that space. So we scrape that periosteum. You know, it's largely avascular, at least at the time of surgery. You have biofilms in there, and not a lot of great access for antibiotics. Very challenging model. Okay, now let me see here if I can write quickly. It's not top secret right now. We actually have this. Oh, okay, don't do that. There we go. So this is the device under development. And what this device does is it controls the release of antibiotic from a reservoir system that has a percutaneous port sticking out of the skin that can be refilled with antibiotic. So you see I can refill this as often as I want or need with any antibiotic combination. Well, there's solubility limits, right? But essentially with any antibiotic or combination thereof that I want to. I can release it to that space. This is a picture of the tissue after that has been in that space. It's apparently appears to be nice and healthy, right? So even this high antibiotic load for 10 to 14 days can eradicate the biofilms effectively while not adversely affecting that space. Now there's a lot of work still to be done here, right? We just have pilot data, but we'll be getting this data over the next couple of years. But this is the bone that's been exposed to those antibiotics. This is the bone exposed to biofilms with no therapy. So this really strong osteomyelitis and sequestriform. What I want to kind of draw your attention to is this graph right here. This is the number of bacteria in our model that we put in at the time of injury. This is the number of bacteria that reside on just that membrane that we put in with the biofilm in the positive controls of infection. The number of bacteria on that peak membrane, that small membrane with the biofilm, even after IV antibiotic therapy, actually slightly higher than the positive controls. And we are effectively able to have a 4-log reduction of bacteria in that site with our device that releases antibiotics. So that 4-log reduction is something critical I talked about on Friday of what the FDA wants to see. They want to see a 4-log reduction of bacteria in a space or site to begin to have this discussion of is it effective, right? But then we have to have that safety factor. Is it safe against those tissues, which is what we're also testing and answering. So I think we're kind of running out of time. Oh, I'm right here at the end. Targeting these biofilms, so checking off all of these different components and aspects that we want to be able to target and hopefully translate clinically some improvement on these, this space of the biofilm-related infection. So a huge thanks to our crew. I mean, we couldn't do this without all of the people in behind the scenes. Fantastic lab, and all of our Curza and Pergo Scientific business people, science people that are all making this happen. So a huge thanks to them, and thank you for having me. I think we have time for some questions.
Any question for us?
Unless I answered everything, that's fine. Yeah, go for it.
So medical devices are made in clean rooms,
Mm-hmm.
I guess to try and prevent biofilms from being there, or for the remnants of them being there when they're implanted.
Sure.
Do you think that is useful, given the infections seem to come from the person, not the product?
Yeah, I mean, I think every step you take to be clean is useful, certainly, because, you know, what if there's a spore there, for example, that you really need to not either have on the implant or eradicate it before it goes into the space. But I think we've maybe thought a little bit too much about that and not enough about the skin, right, or the host microbiome. And so I think we definitely need to make a shift towards thinking more about it.
Spending money in the wrong place.
Yeah.
Trying to keep it clean when actually it just gets dirty.
Yeah, I mean, it's like the contact lens, right? You take it out of a sterile package, put it in your mouth, and then put it in your eye. My sister does it all the time. I don't know if anybody else does. But yeah, I mean, she puts, 1,000 microorganisms right on her eye, right after it comes out of a sterile package. So it's that same concept of, we're making something so clean, but yet we immediately contaminate it with microorganisms. Yeah.
You say the MIC doesn't relate really at all to the anti-biofilm activity.
Yeah.
Wouldn't MBC be a better?
So not really. So the MBC is usually only about 2 dilutions higher than the MIC.
Those are fairly tightly correlated?
They are. So I will say this, though. There are a few antibiotics that do appear to maybe have a little bit better indicator. So not such a massive disparity between the MIC and antibiofilm value. And I think I mentioned this on Friday as well, but that's like daptomycin, rifampin, and gentamicin. So daptomycin, for example, if you run an MIC panel, it's like 2 micrograms per mil is the MIC. If you run an MBEC, the minimum biofilm eradication concentration of the tray, the 96-well plate with the pegs, if you run it against that, I think the MBEC is only about 64 to 128 micrograms per mil, which is still a huge increase, right? Yeah.
But that's kind of the point. The spread between the MIC and an MBEC is unpredicted.
Exactly. Yeah, but as far as, so daptomycin has that, that kind of spread. But if you look at vancomycin, I mean, you're looking at like 1,028 micrograms per mil from 1 microgram per mil. So clinically, it just wouldn't be useful against biofilms. Or we have to dose it for 6 weeks at a time, right? So the question is, over time, does it work, right? How effective is an antibiotic over that length of time, which there are no studies that have tested that, at least on the benchtop. So if I expose a biofilm in a system to vancomycin for 10 days or 30 days, is it going to become effective over time, right? Or is it just completely, consistently knocking back the planktonic bacteria while the biofilm is still growing? So yeah, that's kind of where some of our work is taking us. Yeah?
What's the plan afterwards? So you put your device in,
Mm-hmm.
and then you leave it for a bit. Do you take it out?
Yeah.
And then when you take
Yeah.
it out, that isn't that the same problem as opening up someone's?
Yeah, no, that's a great question. So something, and we don't have time
The
maybe
slides
to.
on my computer.
Yeah, the slides on his computer of the video. I actually have a video showing how we pull it out. So when you deflate it, so you can use a syringe, you can just deflate it, and it collapses down to about a 1 centimeter diameter. And then there's a 1 centimeter slit where that tube sticks out of, and it just pulls through it. So it's a minimally invasive procedure to actually pull it out.
The new prototypes, in fact, I just spent the whole week at the Medical Device Conference in Anaheim working on developing through some features so it fits out through the porthole nicely.
Yeah.
So yeah, that's an engineering feature that is absolutely there.
And one of the things that
We're proud of it.
Yeah. And one of the things that I really like about this is we can put lidocaine in it. So we can actually numb that site before we have to pull it. So it might not have to be an anesthetic procedure. An inhalant anesthetic. We could actually do this in the clinic. So it'd be not as expensive.
And how do you know your biofilm's all gone before you pull it out?
You don't. Yeah, you don't. I mean, you'll still have obviously those problems.
You hope
But
they
at the
cover that.
time of removal, you'll likely do swabbing and see if there's anything still there. Yeah.
The IV antibiotics you showed in the end, they had a worse result than no treatment, right?
Yeah.
Was it, you also found the same with the pig model, I think?
Yeah.
It was treated with colistin or something. You said there was some bacteria, but there was zero.
That's right.
That was right,
Yeah,
yeah.
so with the IV antibiotics, we're finding across our models that they don't eradicate everything, and especially when they're in that biofilm
And the
phenotype.
same with the pig model, right?
Mhm.
It was treated with colistin, was it? Or
Colistin imipenem, yeah. And that was a 2-week course, which is quite a long course.
And that was worse than the control, untreated control.
That's right.
Any sort of rationalization for
Well, so I think you have a couple of things going on. So the eschar, the scab that develops, I think harbors bacteria because we actually have quantified some of the debrided necrotic tissue or even healing tissue, and it still harbors bacteria, and that's avascular. So you're not going to get all of the antibiotic in through vasculature because you're starting to shed skin off of there. And so I think that contributes to it.
the pig bottom one was treated topically?
Yeah, so when you add topical products in combo with IV, then you get the full reduction. Yeah.
So the pig model had only had one combination.
We had both in it, but the one that got IV only still had residual in it. Yeah. Yeah, no, that's something that was very interesting to us.
Completely zero for the immune system on its own, basically, right?
When the planktonic bacteria, yeah. Yeah, that was, yeah, it was interesting. And that's under review right now for publication, so it'll come out soon. Yeah.
You mentioned that the sheep model that was inoculated with biofilms, they didn't need any antibiotics, they wouldn't get that sick compared to the ones that were infected with planktonic bacteria.
That's right.
Can you explain that? Is that like a local response
Yeah.
versus a systemic one with the planktonic?
Yeah, I mean, to the best of our ability to think through it, right? So the planktonic bacteria are highly metabolically active. And so their whole mindset, if you will, is find a place to adhere to and metabolize, right? Grow, 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 eat. But when you have a biofilm, it's already in its state of kind of dormancy, it's quiescent, low-lying, right? You put that into there, and it just kind of wants to hide out. So, and it doesn't necessarily, in this particular model, we don't really have a whole lot of foreign body. We have those silica beads in there, but they seem happy to just kind of stay where they are and not have to, grow and metabolize so quickly. What they do is they just chew away at that bone at a very slow rate. So even at 4 weeks, we really don't even see much going on that bone surface, but at about 8 weeks, it's moth-eaten and osteomyelitic. But if you put planktonic bacteria in there, you get osteomyelitis in about 10 days.
Might even kill your sheep immediately.
Right.
That's the bigger
Yeah.
problem.
And we have to actually give antibiotics to those sheep that we inoculate with planktonic bacteria. We oftentimes have to give a course of antibiotics just to get them through that initial period.
What's really cool is we have a spine model we've developed with a surgeon out of UPIT. And as part of that spine model, we actually give them a full course of antibiotics just to develop a nice biofilm infection. It doesn't even change it just stops any spurious, systemic infections, but it keeps the biofilm just fine.
And so that kind of goes back to that's the reason I showed those 2 early papers of 1982, 1984, the pacemaker and the fracture fixation device, is that the biofilms appeared to serve as reservoirs of infection. So the courses of antibiotics will knock back the planktonic bacteria, but those biofilms serve as a reservoir, right? And so what we're trying to do is, nail the reservoir. Got to get rid of it so it can't seed the infection.
Well, it's close to 5, so why don't we continue our conversation over drinks and nibbles?
Wonderful.
Again, those two
Thank you all very much.
Thank you.
Yeah, thank you for having me.